Okay, so plate readers measuring visible light, UV light, fluorescence, luminescence, or being radiometric measurement. So today we're using a visible light spectrophotometer. So what does a spectrophotometer do? Have you used one of those before? Pretty common lab type instrument, spectrophotometer. So spectrum, the light spectrum, right? So photometer, measuring light, measures the spectrum of light, spectrophotometer. So we'll be using a plate reader that is a spectrophotometer that's measuring visible light. Our same machine has UV light also, so it's out of the visible range, but it's still light. There's infrared light too. That's typically not used though for plate readers, but it has uses. And then fluorescence. You guys remember what fluorescence is? Because next week we're going to be doing all of this stuff with immunohistochemistry. So we're going to be using fluorescent markers. And Brandy, you remember all this stuff. So it has a fluorophore on it. We have these compounds that are good for generating fluorescent light. So the idea is that here's our fluorophore. Light is coming in at one wavelength, and it's going off at a different wavelength, slightly different wavelength. So there's the absorption and emission. Absorption spectrum and emission. And so the wavelength of light that you shine on the fluorophore is slightly different than the wavelength being given off. And the wavelength is determining the color. Your eye picks that up as a difference in color. And so therefore it seems like something glows. It's glowing because the light that's given out is at a different wavelength. And so it looks like the light is actually emanating from the object itself. And that's perfect, right? Because it's one thing to shine light on something and to see the reflected light like we usually see in a room. But none of these things look like they're glowing, actually. But if they could pick up the light at a defined wavelength and then give off a slightly different wavelength, it would look like it's glowing. Have you ever seen black light posters? Like in the hate from the 70s and things like that. The wavelength of light that's given off is different than what's being used to illuminate the poster, and so then you see it looks like the thing is glowing. So that's really nice as a way to identify biological components inside of a cell or something on the outside of the cell, something to do with cells. They're really useful. Okay, so that's fluorescence. And then luminescence is a reaction where there's an enzyme called luminol. And luminol plus ATP is going to give off photons. So it gives off light. It's creating light. Have you seen these globe bracelets and necklaces and things? That's just like a flexible plastic tube, and then you crack it, and you're mixing some solution that's in this clear plastic tube. Have you ever seen this? You can go to this Halloween. You'll see people wearing them. Kids with glowing bracelets and things, and it's got luminol on one part, so it has an enzyme in it that will create visible light if there's an energy supply around, and the biological energy supply is adenosine triphosphate, right? ATP. So that's used a lot in the body to drive all sorts of different biochemical pathways. So when you mix those two components together, it starts to give off light. It's shining. It's giving you, often green and orange are common wavelengths. So you see a lot of that. So we can make use of that too. we run some sort of reaction, and then we might want to measure ATP. I mean, because biological things are making ATP, so it's a good way to get an idea of how much ATP is there in a cell extract, for example. And so you would use a luminescent detection method. So it's a plate reader. These are all plate readers, slightly different. So in the lab, you know, the one that we're using today has the visible light and UV light. On the other bench to the side, we have the fluorescence plate reader. And we have a luminescent plate reader too, right next to this one. It's a small box. So we have that also. We're not doing radiometric. What is radiometric? What's that? Radio waves? It's radiation. Radioactive. So school doesn't have a radioactive license currently. This used to be a big deal decades ago. So when I was doing, my graduate studies, we worked with radioisotopes all over the place. Everything was hot. We were so accustomed to this, we just call it hot. We would have Geiger counters on every bench, and we'd walk around and we would see, oh, look at the corner of my bench is contaminated with radioactivity, and oops, my piece of paper that I had sitting on that spot is now, the Geiger counter would be radioactive, and then, you would touch something and then your gloves would be radioactive, and it got kind of scary. Everything was radioactive. So we did that though because it's a nice sensitive marker. Hard to beat radioactivity. Because we were saying, like the fluorescence, it seems like the light source is coming from the object because of the wavelength given off is a little different. And luminescence, the light actually is being given off in that localized area wherever luminol is, but it's not the object itself, it's wherever you can put luminol. So you try to tag luminol and kind of track something that you're interested in. Radiation, though, it's molecular. You can put in a tritium or a phosphorus 32. These are radioactive isotopes of elements, right? So the actual molecule is giving off energy, and you can track that molecule, that specific molecule. Hard to beat that one. It's really, really a pleasure to work with at some level intellectually, but a nightmare to work with in terms of practically speaking, and having radiation all over the lab, and there are dangers to that. Okay, so that gives you some background. So I started out by saying, we're interested in making measurements. Give me some examples of measurements. Like what would we like to measure? Something you could measure about a cell. What is a cell doing? Well, one of the things was the viability, right? It's just generally, is it a lot? Yes, it's a lot. Then I mentioned ATP is the energy currency of the cell. You could measure that. How much ATP is being produced. So how metabolically active is the cell? So, metabolite. You can measure whether a particular reagent was incorporated. And what I mean by that is, give an example like thymidine. When DNA replicates, it needs the 4 bases. And so one of those, the T for thymidine, is going to be incorporated into the DNA. So if you have a way to measure the uptake of thymidine, then you know how much DNA is being created and whether DNA is being replicated. So you can make a measurement. How much DNA replication is there? You can also have some kind of an enzyme link. So the enzyme link indicators, those work well with the visible light thing that we were talking about. And that is, one way to describe that is colorimetric assay. There's radiometric and there's colorimetric. Maybe just a moment. And then there's the luminescence assays too. That's it's an enzyme, right? The luminol. So you can see that there's, there's other things that you could other ways to break this down, but I just want to give you a feeling for the fact that there's a bunch of things to measure about cells, and there's a bunch of ways to measure those things. And, we got into it a little bit with the cell counting. We have an electronic cell counter, but today we're going to use a spectrophotometer. We're going to be measuring visible light, and the light that we're measuring is linked to the cell viability. So what exactly is it that we're doing today? We've got a nice reagent that's kind of a classic, and it's called MTT. So MTT, dimethylthiazole, diphenyltetrazolium, right? So nobody says that. So we say MTT. Tetrazolium bromide. And so it's a colorimetric assay, though. The MTT assay, there's a color change. And this is classic lab stuff. You guys must have already been through some labs where you've got the test tube and, it goes from pink to orange or something like that, or you get these color changes when you change the pH or something else happens. And so this colorimetric assay is going to measure cell viability. So it's described as a cell viability assay, but related to that, sometimes it's called a proliferation assay too. And you can see where these two things are related. If the cells are viable, are they living or not? Sure. But then are they proliferating or not? Are they growing? And what's the rate of growth? So it's related to those growth curves and cell counting that we were doing. But we can do the same thing using a colorimetric assay, and MTT is one example, and it's the one that we're going to be doing today. So this dye, basically, you start out with a yellow tetrazole. So that's the color of it. We made it up in our cell growth medium, which has got phenol red in it, so you're not going to see much yellow. We could have made it up in PBS, then you would have seen how yellow it is. It's made the red a little bit more orange looking, but so that's what we've got. We've got the beginning stuff, and then that's reduced. So there's a reduction, chemical reduction. What kind of chemistry is that? Oxidation reduction. What's the reduction? How does that typically work? How do you understand what
reduction is when it gains more?
Okay. Yeah, and typically you've got some hydrogens coming in there. Oxidation, there's going to be an oxygen that comes in there. So I mean, it breaks a ring. So the reduction is going to give us a purple formazan. So we're going to be seeing increased purple color. So, what is the, I could draw the chemical compound out. Let's do that, why not? Just to point out that there's, where does the chemistry that you guys are studying, how does that fit in? I mean, we make use of the chemistry all the time. You can just look this up. You get the typical benzene ring type thing, and that's linked to. That's linked to the ring. So you go from that. Also, it's a salt. So there's a there's a bromine ion over here, and in solution, ions separate, right? So you've got one side of the positive charge is going to be in solution and surrounded by the negative ions of bromine, and then you, in the presence of this cellular reductase. You're going to basically just clip this ring here, right here. You're going to break that apart. And by breaking it apart, you're going to change its colorimetric properties, which is why you go from yellow to something that's purple. So it's just the same structure over again. Except now this is broken apart. Yeah, this ring is broken apart. Right? So I mean, that's the great thing about organic molecules is that their spectrum, their spectral properties change based on something happening. And breaking a ring is a big deal. It's going to change the way light reflects off of these compounds. And so, you know, this guy up here is yellow, but this one here looks purple. And it's just because this ring is being cleaved. So the actual place that this is done is in the mitochondria. So there's a mitochondrial. Where does the mitochondria, by the way? Somebody volunteer that? Mitochondria, basic biology. Mitochondria are organelles within the cell that could be described as the energy factory of the cell. It's where you've got metabolism, you've got ATP being created and for the use by the cell. So you're going through these metabolic cycles to generate energy-rich compounds. So this mitochondrial succinate dehydrogenase seems to be the main enzyme that's being that's involved with cleaving this, although sometimes it's described just as a cellular reductase because there's some other dehydrogenases that could be working as well, but it's mostly the mitochondrial one. So therefore, it's showing when MTT is reduced, it's showing you which cells are metabolically active, right? I mean, more precisely, it's like the cells are living. It's a viability assay, okay? But you're adding a pigment, an organic molecule that, when it's cleaved by a dehydrogenase, that is going to be present at a higher level in an active mitochondria. That the pigment will be cleaved and you'll get the color change. So you're actually not just measuring whether it's alive or dead. You're measuring how metabolically active it is too. Cells that are more metabolically active are going to have more active mitochondria, and they're going to convert more of this dye into the purple precipitate that you're going to see. So that's interesting too, and it's always good to really understand what is it that you're measuring here. Just like we started talking about the cell counting, and we're measuring any cell, any particle that goes through the pinhole that has an increased resistance. But that thing, it might be a dead cell. or it might be a piece of crud floating there, right? So I mean, there's some potential for some artifacts just doing cell counting. But here we're actually measuring an enzymatic reaction based on a metabolite inside the cell. So it's a viability assay, a proliferation assay, and a metabolic assay, in a sense. So in order to measure this purple color, you need to be at around 550 nanometers. 570 is ideal, 570 nanometers. How do you get these specific wavelengths of light, by the way? In these machines, I mean, you know, where I'm talking about, you know, a very specific measurement. Why can't we measure this at 400? Well, 400 is like red. So I just said that this is purple, it's not red. So, I mean, at 570, that's purple. So in the spectrum, visible light spectrum, each of these wavelengths has a color. You know, we're describing the color. 488 nanometers we use a lot on the immunohistochemistry, and that's green, right? So, I mean, it's good to sort of remember, you know, that the spectrum is divided into colors, and it's pretty narrow. Visible light spectra is kind of just a couple of different wavelengths. We're within a few hundred, and that's all we can see. You know, somewhere between, you know, 4 and 6. I mean, we don't see very much. So there's lots of different wavelengths in the electromagnetic spectrum. We're only picking up a very narrow range of visible light. So 570. So that we're going to be reading with 570. Where do we get that? So we have a light bulb, a high intensity light bulb. We have filters, actually. So it's like a piece of glass or plastic, and it filters out all the other wavelengths of light, and this is the only one that's allowed to come through. And we have a machine that has a color wheel on it. And so we select the wavelengths of light, and the color wheel turns, and the filter that we want gets into position. You know, I say color wheel, it's a circle with holes in it. And the holes have the filters. And we choose the filter that has, you know, 570 nanometers. And so we're passing light through the one that we want. So the light source here is broad. It's broadband, you know, white light. It's got, you know, lots of different wavelengths in it. And then what's coming out here is 570. Actually, this goes up to about 800. So that's how we can measure that. So this we can do measure absorption, optical density by how much light is coming through at a specific wavelength. So we've got a detector. So you have the filter, and you also need a detector. The detector measures light, or you could say photons. How's it seeming so far? Pretty good? Pretty clear? Okay, so so you guys are going to get a multi-well plate, 96-well plate. It's got some fibroblasts that were plated out on it. The number of fibroblasts is the same in every well. So Thanda did, you know, careful titration, made sure that she was pipetting the same number of cells in each well. So we seeded with 10,000 cells a couple days ago. And so now you've got a confluent small well with the same amount of cells in every well. And then you're going to be adding a test compound, a toxic compound. And the toxic compound we're testing today is SDS. It's a detergent. And it's also, it's the same as sodium lauryl sulfate, sodium dodecyl sulfate, sodium lauryl sulfate, same thing. So if you look in your shampoo or in your soap ingredients, very common to see it there. Toothpaste, you're going to see it there. It's like, wow, so we're using a lot of this detergent. Yeah, it's a very common detergent to use. So we're going to look at the toxicity of SDS over a range. So we want to look at a percent. 1% SDS is definitely toxic. So we're looking at something less than 1%. So somewhere between maybe a half of a percent to, like 100th of a percent, 1,000th of a percent. I don't know. Actually, I can look at the sheet here. Amanda has us doing. Okay, we're doing 0.01. Okay, so we're going between this range, looking at the effect of SDS on cells. So this is pretty relevant. I mean, it's pretty relevant because whether you realize it or not, this morning, if you washed your hands or your face or took a shower, and you were covering yourself with SDS at a certain percentage and leaving it on there for a certain amount of time, and you come into the lab here and we're doing the same thing, but in a controlled way. We're in a scientific lab, we've got plate readers, we've got a controlled experiment, we've got 10,000 cells per well, we're going to add different concentrations of SDS, I think for 10 minutes at these different percentages, and we're going to see how much SDS do we need to kill the cells. Why would SDS kill cells? It doesn't seem to kill much when you're taking a shower. But you don't really have exposed cells either, right? Because you've got the dead squames on the outside of your body, and so the epidermis is pretty protective of your body. You wouldn't want to wash your mouth out with soap, right, Matt? I mean, you don't want to consume detergents generally, right? Because they are toxic and they will kill cells. So we're going to look at how much of the detergent do you need to kill cells, but then realize that the skills that you guys are learning using the plate reader, and there's many different types of plate readers. You're getting your first experience with plate readers today. You're doing a colorimetric assay. That's the color change stuff. If I ask you what kind of assay was done in this lab, it's colorimetric. Is it radiometric? No, that one's radioactive. Is it a luminescent assay? No, that's the one where luminol is used to produce visible light dependent on ATP. So, but we use many of these types of reactions in the lab, and as I pointed out, we had 4 out of the 5 different types of plate readers in our lab right there. So we're very interested using plate readers. It's a great way to generate lots of data. So in order to do these measurements of the percentage, you're going to need to do some dilutions of the reagent. And Fanda said to me, well, I can make up the dilutions to these guys. And I said, well, what about them making up the dilutions, right? Have you done this before? In like a chemistry lab, you make serial dilutions. Have you ever done that? Yeah, maybe a few people. So the idea is you need to create gradations of concentration of something, and to have it be exactly at a certain level. So the way we've got this one set up, we have the 0.5%, and then we've got 0.25%, and then we have 0.1%. Okay, so it does go down. Okay, 0.1%. We have 0.05%, 0.025%, and then 0.01% SDS. So we're looking at this complete range. 1, 2, 3, 4, 5, 6 different points, different concentrations of SDS. And you have to make up this series of solutions. So the way to do that is you make up this one correctly, and then you can just dilute that. If you dilute it 1 part of this to 1 part water, you're going to end up with half the concentration of SDS. So that's pretty easy. Just, you pipette out 100 microliters using one of those little pipetters, and then add 100 microliters of water, and then you'd have half the amount. Then over here, you actually do 1/5 of this. So instead of each diluting, from the previous one, sometimes Fanda jumps back to one before that because it was easier to pipette. So to make 0.1%, you have 50 microliters of this stock solution and 200 of water, and then you're going to end up with 1/5 of this. 1/5 of 0.5% is 0.1% SDS. So you need to make up these solutions correctly. And we might as well do it right on the plate. We've got 96 wells. So you're only using the top 2 rows that has cells on it. So the rest of the plate is free. So on the bottom row, I want you to make up your dilutions of the SDS. If you don't make up the dilutions right, the data is not going to look correct. And we're going to get to the point where we're actually plotting out the data, too. So your curve isn't going to look any good. So, I mean, do a good job on the dilution series and you'll be halfway there. So then you're going to add these SDS solutions to your wells with the cells. Leave them on there for 10 minutes, and then we're going to take that off, and we're going to add the MTT reagent. And we're going to let that incubate for 90 minutes. There's a long incubation in there. During that long incubation, we're going to talk to you about graphing data. So we have the growth curves that you guys have been working on. Dan did a growth curve every day, and you guys did it once a week. So sort of mixed results, but some, you were able to see though that there's some sort of pattern to how the cells are accumulating over time. But in this lab, you're going to be able to see, a reasonable piece of data. So what do you expect to see? Tell me what you think is going to happen. What would the plot? What kind of plot would you have? You have a coordinates x, y. So what are we going to plot here? Well, in the y, this is going to be cell viability. So it's not numbers of cells, but cell viability. And cell viability is measured at 570 nanometers. So these are the reads off of the plate reader. So you're going to be getting numbers. So you're going to plot these numbers here, and then you're going to plot them versus what? We just made the dilution series of what? SDS, right? The amount of it, the percentage of SDS. So we have 0.01, whatever. Different percentages of SDS, and you're going to see that that there's some effect on cell viability. So it's either going to go down or up or something. So you're going to figure that out. You're going to figure out what the curve looks like. Is it a straight line? Is it a sloping? Is it a more logarithmic type of effect? Is there a threshold? Maybe a certain amount is tolerated and then suddenly there's a response to cell viability. So you're going to figure that out. Does that sound good? You excited? Okay, so I think this is a good lab. I really like this lab. This is one of the more popular labs that we've run. It's just because you get a lot of data, because you're able to plot the data, so you're getting a definitive endpoint. You're not just making an observation. You're getting a data set. And you can do it in duplicate or triplicate, because we've got these multi-well plates. So then you have statistically significant data too, which is very powerful. I mean, it's, that's what you need for publication quality data, is you need a statistically significant data set where you can show error bars on your graphs and show people what the response is that you're measuring. So, okay, so that's it. So we can go into the lab and start to work on this. As far as the syllabus goes, by the way, so next week we have the cell imaging. And the following week is similar. We're going to do sort of more complex analysis of the images that we make. And then there's a quiz on that one. So 2 weeks from today, there's another quiz. And then there's 2 weeks that we're off. Remember, there's the fall break and then family weekend. So there's 2 weeks in a row that we don't have lab. So we're kind of trying to pick up the pace here for you guys, because we know that we're going to be taking a vacation there for a while too. So I look at this as kind of the, this is a big chunk of the semester right here. So this lab and the next lab, and then you're off for 2 weeks. Keep that in mind. Okay, thank you.